Okay, apologies for crazy lighting, weird background, and any sounds you might hear. I am staying somewhere right now for somebody's wedding. I am the photographer. This place was supposed to be chill and quiet and whatnot, and apparently a lot of people tend to show up on the weekends. So it's you can tell from the back a little rustic and whatnot. And so yeah. Anyway, first and foremost, my apologies for not being able to make it to class on Monday. I don't think I've covered it in class, but I'm having some different medical thingies, some tests and stuff, and I'm having to get an MRI of my neck. And I'm not real happy about having to get it done because I really don't even know what it entails, other than I'm apparently going to have to take out my 8 million earrings and stuff. So I will be getting an MRI, and then on Thursday I get my follow-up with the doctor, and then the doctor hopefully will have a course of action of what to do. And I have no idea what it'll be. So eventually we'll find out. Anyway, I hate that we're missing this one because this is where we start genetics. And if you remember, I have gone on and on how much I friggin' love genetics. And now the AC unit starts. Sorry about that. I love genetics. It's one of my favorite topics. So I am going to do this PowerPoint and try my hardest to cover everything I normally would cover. And then you have the online lab. There is an old video that goes with the lab, but it still talks you through exactly what to do. And next week, we will then finish up with genetics and do a huge genetics lab in class, and it counts double. So the one for next week is huge. It counts double. It's important. Make sure you're there. All right. Without further ado, let's get to the PowerPoint. So we will do our slideshow. And I do apologize for the randomness of the AC. It is so temperamental. It turns on, then turns off. Sometimes it'll turn off for 2 seconds and then start right back up. But there you go. Okay, so patterns of inheritance. Basically just pointing out that there's some stuff that's passed down through your genes. Here they're talking about in this one diet that's high in protein, very high in fat, consists of food obtained by hunting large land mammals. And what allows these people to tolerate the high levels of dietary fat? Well, part of that has to do with their genes. Here's an Inuit. You may have grown up calling them Eskimos, but it turns out Eskimo is a derogatory term. Inuit is the actual proper name, just so you know. And, haha, genetics. So Mendel, Gregor Mendel, that's the main one we're going to be talking about here. So here we're talking with, I started to just totally butcher this name. Anyway, somebody thought that there were these special pan genes that traveled through the eggs or sperm. Is incorrect because the reproductive cells are not composed of particles, and changes in somatic cells do not influence gametes. The blending hypothesis was rejected because it did not explain how traits that disappear in one generation can reappear in later generations. So they were already way back when on the cusp of understanding genetics. They just hadn't quite figured it all out. So skipping through that. We're going to talk about Gregor Mendel in just a moment. But first, we're going to say heredity, transmission of traits from one generation to the next. This is what you mean when you say, I inherited my eyes from my dad, or, my height from my grandpa, that kind of stuff. It's because it's heredity. It's stuff from your ancestors travels on down. Genetics is the scientific study of heredity, and it began with Gregor Mendel, the one who I keep talking about. We'll have a goofy picture of him in a second. I say goofy because it's like this painting. He looks so serious. What he did, he was a monk and a mathematician, and he crossed pea plants and traced the traits from generation to generation of these pea plants. He's the one who hypothesized that there was an alternative version of genes, which are the units of, or that determine the heritable traits. Not going to make you describe these, but I will tell you P is parent or parental, F is filial, meaning the offspring. And so if the P generation is your grandparents, the F1 is your parents, the F2 is you, F3 would be your kids, so on and so forth. Gregor Mendel. Okay, now, terms used in modern genetics. We have genotype, homozygous, and heterozygous. The genotype is pretty much the exact set that an individual carries. So that is your genotype. The individual with two identical alleles of a gene is homozygous, because if you remember, homo means same. So two identical alleles are going to be homozygous. Non-identical alleles will be heterozygous, hetero for different. Hybrid's the heterozygote offspring of a cross between two individuals that breed true for different forms of a trait. When the individual's genotype, not when, but the individual's genotype will determine the phenotype. So the genotype are the genes. That's like the blueprints. The phenotype is what you observe, what you see, hear, taste, touch, whatever. Phenotype you observe. Any mutated gene is a new allele, whether or not it affects the phenotype. Now, this is one of my favorite hybrids. These are ligers. I talk more about these in Bio 124, but here you go. A liger, daddy is a lion and mommy is a tiger. You may notice that they tend to be monster sized. They are huge. So the large size of a liger and the small size of a tigon is the opposite of the liger, where the mom, oh, did I just mess it up? There we go. Daddy is a lion, mommy is a tiger. So a tigon, mommy is a lion and daddy is a tiger. There you go. So what happens here, why they are, they work. Both have 38 chromosomes, 19 chromosomes from each parent. Both the males are infertile. A true hybrid tends to not quite match perfectly. Something normally is missing, and it normally has to do with reproduction. And so most true hybrids cannot have babies. And then here, talking about techniques for crossing the pea plants. I just skip on over that. He developed 4 hypotheses. There are alternative versions of genes that account for variations. For each character, an organism inherits 2 alleles of a gene, one from each parent. Organism that has 2 identical alleles, homozygous. 2 different alleles, heterozygous. We've already talked about that. Then we have, if the 2 alleles of an inherited pair differ, the one determines the organism's appearance is called the dominant allele, and the other has no noticeable effect on the organism's appearance is the recessive allele. So dominant and recessive, these have to do with the heterozygous part. If you have and you're going to see this in just a moment, we're going to have capital letters and lowercase letters. And the dominant allele is always shown with the capital letter. The recessive is always shown with a lowercase letter. And then a sperm or egg carries only one allele for each inherited character because allele pairs separate. This statement is called the law of segregation. So they separate, so segregation. Here's an example. They took purple flowers. Notice it says true breeding parents. That means everything that came from these purple flowers were always purple. Everything that came from these white flowers were always white. So now they take those and they cross that purple with that white. Well, first generation of hybrids, they're all purple. But then they cross those and now they have 3 purple and 1 white. That lets you know that the white is a recessive gene. And that one is still in there. You didn't see it for 1 generation, but that's because it was hiding basically, or overshadowed. That's another way to look at it. Mendels hypotheses also explain the 3-to-1 ratio observed in the F2 generation. Notice here you have 3 out of 4 were purple, 1 was white. That is the 3-to-1 right there. Now you use a Punnett square to show the 4 possible combinations of the allele that can occur. And you're going to see Punnett squares in just a moment. So here we go. We have the gametes. We have the purple flower has 2 capital P's because it is homozygous, homo same, dominant. So 2 capital P's because all of its babies have always been that. Now we have the white flower, which are 2 lowercase p's. So homozygous recessive. So 2 little p's. Now, each of them can only give 1 P at a time. The purple can only give that big P. That white can only give a little p. That means half of them will have a big P, half of them will have a little p. Since half of them, or since they all end up showing, I shouldn't say half of their genes are big P, half of their genes are little p. So all of them have a big P, which means you can't see the little p. So you see the purple, but you don't see the white. Now you take 2 of the flowers from here, or 2 of the plants. Notice now in the square, let me do the laser pointer. Right here, this is 1 plant. We have a big P and a little p. Here's the other plant. We have a big P and a little p. So you now go, you can go across or down, your choice. I'm going to say across. We're going to take this big P and put it all the way across. And then this little p and put it all the way across. Then we take this big P and bring it down, and we take this little p and we bring it down. Notice this one is now heterozygous dominant. These 2 are homozygous dominant. These 2 are heterozygous, sorry about that. And this one is homozygous recessive, because it got the little p from this one and the little p from that one. And that's what gives us our 3-to-1 ratio of phenotype. The genotype ratio, however, is 1-to-1. This is the same thing, just nice and big, since we talked about on the other one. I'm going to skip on by. Now, every diploid cell has pairs of homologous chromosomes. Diploid means 2. Chromosomes in a homologous pair will carry alleles of the same genes at the same locations. So here we're looking at homologous chromosomes. Homologous, again, same or similar. If you look at these 2, you will see these are going to be showing each one is a chromosome. Each one is showing an allele in around the same place. But notice this one is dominant. This one is dominant. So this one is homozygous dominant. Recessive, recessive, homozygous recessive. Here's the interesting part. We've got dominant and we have recessive. So here is our heterozygous. Cross between 2 individuals that are heterozygous for 1 character is called a monohybrid cross. Mono for 1. So you are crossing 2 individuals that are heterozygous for 1 characteristic, 1 character, 1 trait, one allele. Say it however you want, but it's one. That's why monohybrid, one. Dihybrid cross, two. Mating of parental varieties that differ in two characters. And Mendels' law of independent assortment states that the alleles of a pair segregate independently of other allele pairs during gamete formation. So they are not dependent on something else. Each one can change however it needs to. Here, they're going to show you two hypotheses for segregation in a dihybrid cross. You have this guy. So you've got two big R's, two big Y's. So the gamete can only be a big R and a big Y. This one, the only gamete can be a little r and a little y, which means our F1 generation is big R, big Y, little y. To do the dihybrid cross, you have to have every variation. Now this, I am just going to say, feel free to take a look at this if you want to, but in class when we are covering our big genetics homework stuff, or not homework, lab work, I will draw a giant dihybrid cross on the board for you and work it out. So don't worry, I got you. Independent assortment of two genes in laboratory retrievers. Black coat, normal vision. That is the dominant allele. That's why you see a big B and a big N. And those blanks mean it doesn't matter what those others are. Black coat and blind means two recessive. So homozygous recessive for this genotype. Chocolate coat, again, homozygous recessive for that. And chocolate coat and blind, all recessive. And this will give you a ratio of 9 to 3 to 3 to 1. And again, we cover this in class next week. Offspring of a test cross is a mating between an individual of unknown genotype and a homozygous recessive individual. This is how you can figure out the unknown genotype. So like with the pea plants, if you know that the white flower is the recessive, you can cross it with any of the others. If you get any white at all, then you know that it was a hybrid, heterozygous. If you cross the purple with a white and you get only purple, then you know that first one was homozygous dominant. Hope that makes sense. I know it gets confusing. We will totally cover it more in class on, the next Monday. This just showing you again. Rule of multiplication calculates the probability of two independent events, both occurring. And the rule of addition calculates the probability of an event that can occur in alternative ways. Will I make you do this? But we'll cover it next week if you want to. They like to have you do this to kind of figure it out. I'm not making you do this because we do similar in our lab next week. Inheritance of many human traits follow the Mendels laws, and family pedigrees can help determine an individual genotype. They use pedigrees for animals as well, but a pedigree for a family history is actually how they can predict certain disorders. And so that's a good thing to look for. This one's talking about straight line and a widow's peak, dominant recessive. We had a whole other lab we could have done with this, but you have the virtual lab this week because of what's going on. This is an example of a pedigree where it's showing you females are circles, males are squares. If it's an empty square or circle, then that means they have the dominant trait. If it is colored in, they have the recessive trait. They also use these for tracking diseases. I kind of find these interesting, and I have had fun with these in the past, but I don't make you do this, just so you know. Okay, we have some genetic disorders that we will be covering, and sometimes you will have parents who appear perfectly normal, like they don't have anything wrong, and it turns out they're heterozygotes for a particular disorder. And if they're heterozygotes, oops, for a certain disorder, then they can be carriers of the recessive allele. That's why they can have a baby that's affected. A couple of weeks ago, I taught you about Tay-Sachs disease, and that is a perfect example. No one with Tay-Sachs lives. They die by the time they are 5 or 6 years old. So you are never going to have somebody who is afflicted with Tay-Sachs have a child. But Tay-Sachs keeps continuing because people still carry the gene. So if you take two parents who are heterozygous for the Tay-Sachs gene, and then they have babies, each time they have a baby, there is a 1/4 chance, a 25% chance that the baby's going to have Tay-Sachs. So that is what is being a carrier. Here's just a list of some different disorders that you can pass on through your chromosomes. We also have freckles and albinism. So freckles are a dominant trait. No freckles is recessive, and we will totally talk about this one in lab next week. Normal pigmentation, which is most people you see, or albinism. Albinism, there's no pigmentation, so it's completely white. Again, here's another Punnett square to show you how to do this, but trust me when I say we will do mounds of these next week. Now, carrier screening, fetal testing, fetal imaging, and newborn screening can provide information for reproductive decisions, but may create ethical dilemmas. What does that mean? It means they might think there might be an issue, and so they can do a test. They can do a few different tests, and we're going to talk about them in a second. Here we have the CBS. This allows the genetic screening to be performed very early in pregnancy and provides quick results, but it's invasive. And so you might be able to find out that there's something wrong and have a way to fix it. Because like there's a certain one where when you get it tested, you might find out that the baby is intolerant to a particular something. You can alter the mom's diet and you can totally fix the problem. So that's a really good reason to do it. But since it's invasive, it carries a risk of causing a miscarriage. So you might lose the baby. Other times people test for things like Down syndrome. And then that's where the ethical part comes in, because if you test and you are told that the baby you're carrying, or your wife is carrying, your girlfriend's carrying, whatever, if the baby that is in utero tests positive for Down syndrome, some people will choose to terminate the pregnancy. The tests are usually pretty good, but they are not foolproof. And here's where I let you know about a friend of mine who she already had 2 little boys. She was pregnant her 3rd time. And they told her that they suspected the baby had Down syndrome. She had a test done. They said, your baby's going to have Down syndrome. And she just started researching and preparing. And all these people kept coming up to her asking, what are you going to do? And she's like, what do you mean? What am I going to do? I'm having my baby. She was really upset, and, rightfully so. She had her baby. Beautiful little girl. Did not have Down syndrome. So while it might help you sometimes, it's not always 100% accurate. So you might need to do more tests down the road. Just a heads up. Now, in humans, we have 2 sex chromosomes. Human males have an XY. Females have XX. Yes, you need to know this. So men have an XY. Women have XX. All of the other chromosomes are known as autosomes. So the sex chromosomes are the XY or the XX. All the rest of them, are autosomes. So, sex determination in humans. Sex of a child is determined by the father. Let that sink in. It is determined by the guy, because mom can only give X's. Take a look. Here's the egg. two X's. Moms are XX. That is their genetic code, XX. So when they make an egg, it has an X and it has an X. That's all mom can give is an X. Then we have dad. In the sperm, we have an X and a Y. So dad can give a Y or he can give an X. Now you do the Punnett square. Here's mom. She can only give X's. Here's dad. He gave an X here, but he gave a Y here. 50% chance of having a girl, 50% chance of having a boy. That's the way it works. Position, time of year, all of those things, none of those help determine whether or not it's going to be a boy or a girl. None of it matters. It's literally this. It's the luck of the draw. The reason I like to make sure to point out that the sex of the baby is determined by dad is because if you look through history at how many women were persecuted, executed, beaten, destroyed, and whatnot, because they quote unquote did not give a son. Not her fault. If a king kept having little girls, it's because that's what his body was doing the best of. It's never been mom's problem. I mean, well, it was always her problem, but she couldn't fix it because she can only give an X. My throat's bugging me, so sorry. Many human traits can be traced to autosomal dominant or recessive alleles that are inherited in the Mendelian patterns. And some of these alleles will cause genetic disorders. So, dominant autosomal allele is expressed in homozygotes and heterozygotes. This is the one you're going to see. So it says it tends to appear in every generation because it's dominant. With one homozygous recessive and one heterozygous parent, children will have a 50% chance of inheriting and displaying the trait. Achondroplasia is one way. It was one of these, and Huntington's disease is another. I should point out achondroplasia is a type of dwarfism. I think we have a picture in a moment. Autosomal recessive alleles expressed only in homozygotes. The heterozygotes are carriers, but they don't have the trait. So that means a child of two parents that are carriers, there's a 25% chance of the kid having the trait, like when I talked about Tay-Sachs, or this condition right here. Okay, this is achondroplasia. Words are hard sometimes. So here it's showing you the affected father, and that is a dominant allele. And so you see he is affected, he passes that on. Then you have the other case where if it's the recessive trait, both mom and dad have to carry the trait in order to have an affected child. Progeria. This one is fascinating and terrifying. This is a hyperlink here. Let me turn off the laser pointer so you can see. Notice that is a hyperlink. So if you want to hear or read more about this disorder, click it. What happens here is it causes a person to age at a massively accelerated rate. If anyone has ever heard of, it's something like a dahlia rose, but I don't think I got a dahlia right. It's something very close to that. She has actually survived longer than a lot of people with progeria. She's got a YouTube channel and stuff like that, and she does art and stuff right now. So this one is fascinating. I highly recommend you check it out. Okay, examples of X-linked inheritance. These are showing up on the X chromosome. Mutated alleles on the X chromosome cause or contribute to over 300 genetic disorders. The X chromosome is huge. It holds so much information. More males than females will have an X-linked recessive genetic disorder. because since males only have one X chromosome, if it's on it, it's going to show. Female has two X chromosomes. So if it's only on one of them, the other one is going to hide it. There you go. Again, just trying to show you how it works. Some X-linked recessive disorders, we have hemophilia A, otherwise known as like a free bleeder, red-green color blindness, and muscular dystrophy. Now, this one is always better in class because then I get to say, okay, take a look. The rectangular pictures, it's showing you one that is definitely red and green. The other picture is supposed to show you what it would look like to somebody who might be red-green colorblind. The ones labeled A and B, I find those the more interesting ones because more often than not, when we do this in class, at least one person in the class won't see one of these numbers. So I love to have you guys just check it out and then see if you can see the numbers in A and see the number in B. Let me know. If you couldn't see them, let me know. I'd love to know. Okay, on rare occasions, chromosome structure changes. And these changes usually are harmful or lethal. Very, very rarely are they neutral or beneficial. What can happen here is that a segment of a chromosome can be duplicated, deleted, inverted, or translocated. So here's an example of duplication. The normal chromosome, we just have A, B, C, D, E, F, G. Something goes nuts in prophase 1, and now we have A, B, C, D, E, D, E, F, G. That's going to mess something up. Deletion. This is where one part just gets deleted somehow. Example for this is CredaShot. This is a hyperlink about CredaShot, also known as Cat's Cry. And this is a video that I would love for you to watch. I'm not going to watch it on here because I'm probably going to have to upload this one to YouTube. It's going to be too long. And then if I play this, it's probably going to, tag it for having music or something from someone else. So I'm just not going to do it. But if you download the PowerPoint that's already there, in week 6, it says my PowerPoint, open it up, says my chapter 9. If you download that PowerPoint, you can then watch this video because the video is embedded in the PowerPoint. I highly recommend you do. It tells you all about CredaShot syndrome. Very fascinating. Okay, inversion. Again, something just gets flipped. That little flip changes everything. Translocation. It goes from this chromosome to a completely different chromosome. It's like, it's not, this causes a lot of issues too. Does the chromosome structure evolve? Well, changes in the chromosome structure can reduce fertility in heterozygotes. But the accumulation of multiple changes in homozygotes may result in a new species. This is how stuff happens, and we cover more of this in Bio 124. Okay, now sometimes a new individual, baby, ends up with the wrong chromosome number. And these consequences can be minor all the way up to lethal. So we have aneuploidy, so too many or too few copies of one chromosome, and polyploidy, poly for many. So polyploidy is 3 or more copies of each chromosome. Changes in these chromosome numbers can be caused by non-disjunction. This is when a pair of chromosomes fails to separate during mitosis or meiosis. At this point, I'm thinking, I hope you guys watched the chapter 8 video. If you did not, make sure you do, because you need to understand mitosis and meiosis for this. And I'm trying to make a mental note. We will discuss those in class on our next Monday as well. Please don't let me forget it. I will draw mitosis and meiosis on the board because you will have to be able to draw and label one of those on your test, as well as do this genetic stuff. Okay, there we go. All right. affects the chromosome number at fertilization. So non-disjunction means during meiosis, when it's separating like this, well, the chromosome's getting pulled. Sometimes they don't get pulled evenly. That's what non-disjunction means. So then you will end up with monosomy or trisomy. Monosomy, n-1, and trisomy, n+1. So here's non-disjunction that I just tried to demonstrate with my hands, but there you go. Notice anaphase 1, when they're supposed to separate properly, they didn't separate evenly, which gave you too many chromosomes in metaphase 2. Anaphase 2 separates nice and normally. However, you have too many chromosomes in that cell. So only trisomy 21, which is Down syndrome, will allow survival to adulthood. So trisomy 21 is Down syndrome, and it is caused by non-disjunction. I'd remember that, just so you know. Incidence of non-disjunction increases with the maternal age. Here is a karyotype of someone with Down syndrome. And I'm going to do the little laser pointer again. So notice as you're going through all of our chromosomes, then there's 23 pairs of chromosomes, or 46 chromosomes. That notice they all are very similar. So that's how they're matched up. All of these are autosomes. These are the sex chromosomes. That is your 23rd pair, sex chromosomes. That is an X and X. So this is a little girl. This trisomy 21, you see 3 chromosomes on the 21 mark. That means Down syndrome. And here's where it's talking about Down syndrome and maternal age. The older a woman is when she gets pregnant, the more likely Down syndrome is to take place, because as the woman is aging and her eggs are aging, non-disjunction is more likely to occur. There is also Turner syndrome, tends to be from the father's age. The increased father's age tends to cause more cases of Turner syndrome. Ah, there it is, Turner syndrome right there. So changes in the sex chromosome number can impair learning or motor skills or be undetected. So female sex chromosome abnormalities. We have Turner syndrome, which is shown as XO. It's basically missing a sex chromosome. So there will be just an X. That's it, just a single X. Then we have triple X syndrome. And triple X syndrome is 3 or more X chromosomes. Male sex chromosome abnormalities are Klinefelter syndrome, which is XXY, and then the XYY syndrome. So Turner syndrome, again, usually caused by non-disjunction in the father. This results in females with undeveloped ovaries. Yeah, that's, I guess, all the information I have on that. They also tend to be a little smaller in stature and probably don't have normal periods. They may have a period, but it's not normal, and it's not likely for them to have children. So because of the undeveloped ovaries. Triple X syndrome, trisomy X or 47, triple X, whatever. This just means that there's an additional X chromosome in each of the female cells. These women might be a bit taller. They might have some small issues, blah, blah, blah. It's, some basic things. A lot of it, I think, seems to come along with standing out, But there are some abnormalities, but nothing massive. Klinefelter syndrome, XXY. This one always breaks my heart, because this is that guy who probably is made fun of a lot in middle school or high school, because his body proportions don't match a typical boy. A typical guy tends to be wedge, broader shoulders, narrower hips. And a gentleman with Klinefelter syndrome tends to have shoulder and hip size about equal, which is more of what a girl has. That's why girls are the hourglass. They also tend to have abnormally large breasts, gynecomastia, but man boobs. You've heard people go, oh, look at his man boobs. Sometimes they can't really help it. And it's honestly for this: infertility, sexual problems, less than the normal amount of body hair, pretty much. You know, these poor guys get picked on a lot, and it's nothing they can help. It is literally their chromosomes. Then we have XYY. This one cracked me up. For a while, people were thinking that they had found the criminal gene. And what it turned out to be was a lot of these guys tended to be taller. And if, say if you got robbed and then there's a lineup and the guy who robbed you was 7 foot tall, you're probably going to remember what he looked like. And that's what it was. Once they started actually searching and researching it more, they found out there was no higher rate of criminals with XYY syndrome than there were with people without it. It was just, since they were so tall, it was just easier to catch, easier to spot. That's all. Genetic abnormality. This is a rare or uncommon version of a trait, not inherently life-threatening. Then we have the genetic disorder. The disorder is an inherited condition that causes mild to severe medical problems. And it's characterized by a specific set of symptoms. And this is called a syndrome. So here's just a table of a whole bunch of these if you feel like taking a look. We have recurring genetic disorders. Mutations that cause genetic disorders are rare and put their bearers at risk. These mutations survive in populations for a few reasons. They can reintroduce, or they're reintroduced by new mutations. Recessive alleles are masked in the heterozygotes, and the heterozygotes might have an advantage in a specific environment. One of the ones I will talk about next week, if I don't do it in this one, will be sickle cell disease. Sickle cell is one of those that you can't fathom why it should be here, but it actually gives an advantage to a certain region. So we'll talk about that soon. Genetic counseling starts with the parental genotypes, pedigrees, and genetic testing for known disorders. Information is used to predict the probability of having a child with the genetic disorder. So they do tests. Here's where I said we were going to talk about the different parental tests. Here we go. They're going to do the tests on an embryo or a fetus before birth to screen for sex or genetic problems. These can involve risks to the mother and the baby. Yes, I know it says the fetus, but babies. I love babies. So 3 types of prenatal diagnosis. We have amniocentesis, we have the CVS, which is the chronic villus sampling, and we have fetoscopy. When a severe problem is diagnosed, some parents choose an induced abortion. They choose to terminate the pregnancy. Other times, however, like I mentioned before, it could be something that is 100% doable. Like as soon as you have the knowledge, you can do something to fix it. PKU is an example. It is a very rare condition where the baby's born without the ability to properly break down this one amino acid. However, if none of this is fixed, then this can cause central nervous system and cause brain damage to the baby. But I didn't put it in there, but if you treat mom and she makes her adjustments in her diet and she does everything she's supposed to, they can fix this in the baby before the baby's ever even born. So you also need a mom who cares more about baby than she does about what she wants. And that's a whole other story right there. Genetic screening, widespread routine testing for alleles associated with genetic disorders. This is so you can have a clue if you're likely to possibly have one of these issues. So if you have family members with some kind of genetic disorder, syndrome, whatever, chances are you might pass that on to your kid. So different things like that. Amniocentesis. Here we go. This is the amniocentesis. They take a needle, they put it in here into the amniotic sac, and they pull out the fluid. They then do the centrifuge, and they check the fetal cells, the baby cells. They culture it, and then they can do it, and then they can do a karyotype so they can see how the baby is. And look, this one is a boy because that is XY. And then the CVS, it is a little more in depth. They're using the ultrasound. They put the tube up into the uterus and take it from there. That is why this one can sometimes cause a miscarriage. I just completely blanked on the word. And here's just an ultrasound. So you get to see the baby in there. And anyone who's ever been present for one of these, it's so cool. If they have the sound on, you get to hear for the heartbeat. It's pretty cool. This is a video. Let me turn off this so you can see. This is a video so you can actually see them doing an ultrasound. So if you've never seen one, it's pretty cool. But again, you'll have to download the PowerPoint to get to it. Now we have some variations on Mendel's laws. These we are going to go into so much detail again next week. So I might go a little fast. If so, my apologies. We will cover it more. All right, we have complete dominance is what we've been looking at. But sometimes we have incomplete dominance. So here we have the big R and the little r. But now instead of having 4 red ones, we have 4 pink ones because it's blended. So it's either an incomplete dominance or blended dominance. You pick which phrase you want to use. They're both the same. Now all the pink ones have both the big and the little r. So then if you breed these two, you will get 1 red, 2 pink, 1 white. This is still 1, 2, 1. So the genotype is the same as, or the genotype ratio is the same as it was in the other monohybrid, but the phenotype ratio is different. Co-dominance, I say co-dominant like co-captains. Both of them are fully shown. You see them completely at the same time. And again, totally covering this in our massive lab next week. And then we have the multiple allele system. This is going to be your blood type. So remember, the genotype is the gene, it's the blueprint. The phenotype is what you observe. So if you have the phenotype A, your genotype can either be AA or AO. Phenotype AB is genotype AB. That's all there is to it. Phenotype O is genotype OO. Phenotype B is genotype BB or BO. And yeah, we'll cover this again next week too. I don't make you do this whole thing. I just talk to you about it a little different. So we'll just skip on past that one. And go back there. Pleiotropy occurs when one gene influences multiple characters. And sickle cell disease, human example of pleiotropy. So a moment ago, I talked about sickle cell and how you would have to wonder, how could there be anything good for this? Well, if you think, let me see, what's my next slide? Nope, it doesn't show up. For sickle cell, if you think of the area, or shall we say the ethnicity that tends to have a higher rate of sickle cell, you are going to probably say people of African descent, and you would be correct. If someone is of African descent, then let's go ahead and just say, hey, from Africa. Africa, there is something else that kills a lot of people. That would be malaria. So what happens here is malaria is basically a disease based on the life cycle of this plasmodium. The plasmodium is inside the mosquito. The mosquito bites you and injects the plasmodium into your bloodstream. It goes through your bloodstream into things like your liver and your kidneys, since that's what cleans your blood. And that's what helps the plasmodium with its life cycle. And it has all its babies and it's filling up your blood. And then you get malaria and you die. Meanwhile, another mosquito bites and picks up the new guys and takes them on to someone else. Now, if you have sickle cell, some of those nice big plump blood cells are not like this. They're sickled. Plasmodium can't travel on that. And so it's like birth control to this plasmodium. And so if you have full-fledged sickle cell, that's not good, because that causes so many other problems, with organ death being part of it. We talk about that later. We definitely cover it in Bio 124. But if you are a carrier of this gene, that means some of your cells sickle, and that means that you are more resistant to malaria, and so you are more likely to survive. So pretty cool, right? Okay, here's another one. We have what's called Marfan syndrome. Some people will have pleiotrophy, which is where this gene influences multiple traits. So we have some tall, thin athletes that have Marfan syndrome. Think about way back when. I don't think it happens nearly as much now, but you will have that incredible, insanely tall high school basketball player, star athlete. He's playing and then he just collapses in the middle of a game. And you're like, oh my gosh, what happened? If he had Marfan syndrome is a disorder of the connective tissue that includes your heart, your whole cardiovascular system. I mean, it's not just that, but that's part of it. And that's a big deal. So that's what happens sometimes. Okay, here's the sickled cells that I was talking about. Already talked about it. Oh, here we go. We've got polygenic inheritance. So this is a single phenotypic character that results from the additive effects of two or more genes on a single phenotypic character. So human skin color, that's an example. We have so many variations. That's why I don't care if you are as black as can be or as white as can be. You have relatives and whatnot, and you will see all of you have slightly different shades. It's because it is so complex. And that's what all this stuff's about. And then the book decides to show some people's arms to show you differences in skin color. In our classroom, you can look around. We see different. This picture drives me nuts. I really think that one of the authors just wanted to show pictures of his daughters, you know. Okay, chromosome theory of inheritance. This states that a gene occupies a specific position on the chromosome, and chromosomes undergo segregation and independent assortment during meiosis, which we've talked about before. Okay, so this is just showing you how it can separate as it goes on. We've got meiosis 1, meiosis 2, and it just shows variations. We already saw this dihybrid cross earlier. We looked at the Punnett stuff and it said that this one did not show a 9:3:3:1 ratio. What they found was an example of what's called linked genes. So linked genes are ones that are so close together on the chromosome, excuse me, that they tend to go together. It's not that they have to, it's just in prophase 1 when crossing over occurs, these guys are so close together, it is very rare for them to get separated. Yeah, this is all pretty much the same stuff we've been talking about. Not going to these details. Don't you love when that happens on these PowerPoints? Okay, sex chromosomes and sex-linked disorders, right? Genes, sex-linked genes. All right, we've already said XY for a boy, XX for a girl. The Y chromosome has genes for the development of testes, whereas an absence of the Y allows the ovaries to develop. Human males and females both have 44 autosomes, and then they have the 2 sex chromosomes. Some animals, the environmental temperature will determine the sex. So for some reptiles, temperature at which the eggs are incubated for a very specific time are going to decide if it's going to be male or female. This is causing some issues because of our current climate crisis and as things are warming up. This is actually messing up a lot. That's just the XY system, linked genes. Already covered this, not going to worry. Epistasis. 2 or more gene products influence a trait. Typically, one gene product suppresses the effect of another. So, coat color in dogs. Here you go. If there are homozygous recessive, it's basically turning off the color coat. That's how you get your yellow lab. Yeah. Most X-linked human disorders are due to recessive alleles and therefore are seen mostly in males. Kind of like what we already talked about, that the boy only gets an X from his mom. I mean, yeah. A boy only has one X, so it's going to show up. Here is another pedigree. This is about hemophilia in the royal family of Russia. So remember, the circles are girls, the squares are boys. If it is clear, then it is normal. If it is filled in, then that means that they are afflicted. If it's only half, that means a carrier. So notice all the women down the line, all the women were carriers. But the boy, the son, ended up being afflicted. Okay. Here's where they're just saying that they've discovered that, well, in 2003, geneticists discovered that about 8% of males currently living in Central Asia, have the Y chromosome that's very, very, very similar. What they have discovered is Genghis Khan had, as he conquered and pillaged and raped, he had impregnated so many people, so many women throughout this time that his gene is still 8% of the males have part of him in them. Now we get to our nature versus nurture, and I swear we are getting close to the end of this. So if you've held on this long, whew, good job. Variations in traits are not always the result of differences in alleles. Sometimes it's the environment. So here I want to make sure you understand genotype plus environment equals phenotype. This is important. This will 100% be on the test. So genotype plus environment equals phenotype. This means you have to already have the gene in you. So you have this gene and then something happens in the environment and it will make a different phenotype show. Environmentally driven changes in gene expression patterns can be permanent and heritable. So it can be passed on to your children. Here's the first one I always use as an example, is the snowshoe hare. And yes, on the test, I will even ask you for an example. So the snowshoe hare. Most people, when they hear snowshoe hare, they think of just the white one. Well, that's winter time. When it's cold, the snowshoe hare has white fur because if it didn't, more predators would see it and he'd be munched on. So he has the gene to make this happen. The environment gets cold. So his phenotype is white. Now, as it warms up and the snow is gone and you've got grass and trees all around you, if this white bunny was jumping around in the grass, it would get munched on then. So as the environment warms up, the gene in this bunny goes ahead and changes the phenotype. He starts to lose his white fur and have it replaced with brown. Helps him camouflage. So that is one of the examples of genotype plus environment equals phenotype. Another example, I always use this, has to do with drugs. When I talk about weed, marijuana, I know there's a lot of people who are like, hee, 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 hee. I always point this one out. Anyone who has been around people smoking it, you will note that there is almost always at least one of those people who's like, what's that? Did you hear that? They start to get paranoid. These are the ones you need to really watch out for because, and I'm not saying it happens to everyone, but this is an example of genotype plus environment equals phenotype. Some people are predisposed to particular issues, which is why our next step is mood disorders in humans. Some people have the genes that they are borderline schizophrenic, they are borderline bipolar, they are borderline this, borderline that. It's not showing, it has not been the phenotype yet because it's just right here. In some of these people, when they start to get high, when they smoke weed, it's tipping a scale. That is the environment adding something. And a lot of these people, it will flip over. And so someone who is not quite there, smoking marijuana, the THC, can flip that switch, and then the phenotype will be the mood disorder. It will be the schizophrenia. It can be the bipolar disorder. It's terrifying, but it's real. And so it's not just, for whatever. That is an example of genotype plus environment equals phenotype. So that's how we get to our mood disorders in humans. Environment is a factor in schizophrenia, bipolar disorder, depression, and a whole bunch of other mood disorders. Stress-induced depression causes the methylation-based silencing of particular nerve growth factor, and some antidepressants work by reversing this. So if somebody is clinically depressed, I don't mean situational depressed, like if your dad died, yes, you're going to be very, very sad. I don't think you typically need medication for that. Some therapy would do you good. If you are clinically depressed, and I have a friend like this, she has to take her depression meds every day or everything's off. It's kind of like somebody who is diabetic. You don't yell at the diabetic for taking their insulin because their body is not producing the insulin. Well, if they're diabetic, diabetes type 2, that means their body is not receiving it and they have to do something else, but you get the point. Same thing for the antidepressants. Now, what normally happens in class when I talk about this is somebody will tell me somebody they know was on antidepressants and it never did any good. What often happens with these mood disorders, depression, schizophrenia, and bipolar, a whole bunch of these different types of mood disorders, the medication is trying to balance their body back to where it needs to be. It's trying to fix the chemical imbalance. But then these people will smoke weed, which will mess it up. They'll drink alcohol, which messes it up. They'll smoke cigarettes, which will mess it up. Yes, nicotine can knock some of these medications off. So if you know someone who is on medication for a mood disorder and they're saying it doesn't work, make sure that they are being 100% honest with their doctor about everything they're doing, because mood disorders also have a tendency to make people to try to self-medicate, which is when the drugs come in, the alcohol comes in, the cigarettes come in. All of that is a person trying hard to fix what's wrong, but in reality, they're just making it worse. So I have so much I can say about this, and I was going to, but since I'm not in class and I don't really feel like putting it on record here, if you want, ask me questions in class. I'll give you a lot. I have had way more experience with this lately than I would ever care to. So, exceptional creativity often accompanies neurobiological disorders. So, neuro is brain, biological disorders. So, this could be schizophrenia, autism, chronic depression, bipolar disorder, that kind of thing. Most of these neurobiological disorders do not follow a simple pattern of Mendelian inheritance. It's not as easy to track. So it's a little more difficult, but that could also be due to different severities. And again, the environment really, really, really plays a part. The schizophrenia is a hyperlink in case anyone's interested in that, because this was an article that was written a few years back that shows that schizophrenia is actually a whole bunch of different things all put together. Autism, clearly the disorder of my heart, because I told you on day one that my youngest, my oldest, and myself are all on the autism spectrum. So of course, this is going to be my big one. This is the test I took quite some years ago after my youngest was diagnosed. And I was like, because I met this guy named Tony Atwood, and he kept calling me tree. And I asked him, why are you calling me a tree? And he said, because the apple usually doesn't fall far from the tree. And I was like, And he said, so let me ask you something. I said, what? And he goes, do you teach math or science? He didn't even know I was a teacher. And I'm like, science. Apparently, that's a very aspy thing to do, is to teach math or science. And anyone in class who has paid attention, I've offered to help tutor you in math because I like math. So thought it was interesting. But I thought you might get a kick out of seeing how my brain looks. So there you go. Now we are done with chapter 9. There's a whole lot you should be able to do now. Okay, so now I stop the share and we wrap this up. Took forever. Oh my gosh, it was long. My voice is like, and I'm finished. I am sorry I'm not there to do the class on Monday, but I will be 100% the following Monday. And if you have any questions, jot them down, email me, ask, or write them down and ask me in class on the next Monday. I would tell you what day it is, but I don't have my calendar on me. I just know it's not the 20th. So yeah, that's it. Don't forget to do your lab. Don't forget to get your scientific method assessment turned in. I will be grading things as fast as I can, so then you will know right where you stand. And I guess that's it. So peace out.